Okay, so I think we're live. I can see somebody viewing it already. I think it's you, right?
Hindi pa po.
Di pa ikaw yun. Anyway,
Ay,
it's
ito na po. Okay.
ayan na. Ayan na. I think that's you
Opo.
na. Okay,
Opo.
so we're live. All right. So basically that's the difference between pathogenicity and virulence, right? So the degree at which ability to cause a disease and the degree of pathogenicity. Okay? Okay. So it's basically both level and it's basically both ability and degree. All right. Now, let's talk about general examples of pathogenic determinants. As I mentioned a couple of seconds ago, it's by either adherence factors, antiphagocytic factors, enzymes, and toxins. So we're going to talk about everything one by one. Pili. Sorry. Pili is adherence. So basically, its ability to stick through surfaces, particularly cellular surfaces. Antiphagocytic factors, from the name itself, it prevents or it inhibits phagocytization. And we're going to talk about toxins in the next slide. So, toxins by nature is different based on the source. So when I'm saying the source, it's either a gram-positive organism or a gram-negative organism. So exotoxins and endotoxins are the two things that we're looking for. An exotoxin is released mostly by gram-positive organisms and gram-negative organisms, while endotoxins are gram-negative only. Okay? So exotoxins exit out of the organism. It goes out of the organism or it's released by the organism. While an endotoxin, the organism itself could be the pathogenic determinant that's causing the problem. Okay? Its adherence to a specific part of the body can be a toxic activity by itself, or it could be a toxic process by itself. The structure of the cell could be toxic in it by itself. Okay? So that's why it's only released by gram-negative organisms only. So be mindful of this one. This one is a high-yield question. I want you guys to put a highlight on it because this will be important even when you go to your actual practice, either as physicians or as medical technologists. You will come back to this slide and you say, "Oh my gosh, Sir Manuel is correct." Gadard. Okay. Now, let's talk about differentiation of the types of bacterial toxins based on molecular composition. So obviously an exotoxin is released, and the only way it can become toxic to the human body if it's in the protein form. Means to say that it doesn't require a carrier molecule. Okay? So an exotoxin could be protein based or peptide based if it's connected to other proteins. And an endotoxin is lipid in composition. The example of which is lipid A. Okay? Okay? Clear? Okay?
Yes, sir.
All right. Now let's move on to the next one. The release mechanisms of endotoxins and exotoxins. So exotoxins, as I mentioned before, are either metabolic products or are released as the organism metabolizes something or through treatment. Okay? Whilst an endotoxin is released by the disintegration of the cell wall or by phagocytosis. Okay? Or phagocytosis rather. Phagocytosis tuloy. Phagocytosis. Okay? So it got destroyed in the process, and hence the parts of the organism is actually considered as immunologic or could cause toxicity to nearby cells where the organism has colonized, or it can cause problems with the cells that are phagocytizing it. Could be a monocyte, it could be something, it could be a monocyte, it could be a neutrophil, okay, or it could be a dendritic cell. Somewhere there it can cause a problem. Now we're going to talk much about the mechanism of every organism that we will discuss, medically important organisms that we have. So I'm going to leave it at this. This one is released by disintegration. Okay? So those are the difference. So exo nga parang lilabas lang niya. Exo, nasa loob niya. Pag nasira siya, saka siya, saka siya magiging toxic. Okay? Clear?
Yes, sir.
All right, wonderful. Now, which bacterial toxin type is heat labile and which one is heat stable? So which one? So we already have a basic understanding, right? Exotoxin is a protein. Endotoxin is lipid. Which one do you think is much more stable when heated? Sige.
Yung endotoxin po, yung lipid.
Endotoxin is much more heat stable. Thank you so much.
Yun
All
po.
right, wonderful. Very good. Now, what about protein? The reason being is because it's protein. It can be coagulated or disintegrated by heat, right? Cool.
Yes, sir.
Now, what is the reason why it's important for us to talk about the heat stability and the heat resistance of a particular bacterial toxin? Because when you look at the human metabolism or human physiology, one of our defense mechanisms is to combat a disease is through having fever, right? Nakita niyo kung gaano kaganda ang katawan ng tao, 'di ba? Enjoy niyo 'yon. So kapag ang tao hindi nagkakaroon ng fever, kawawa ka. Kasi ibig sabihin, hindi mo na di-destroy yung toxins, di ba? Kaya pagka ganon, go, go, go ang katawan, sige lang fever ka lang. Pero wag umabot ng 41 kasi magkakaroon ka ng convulsions nun. Okay? Okay ba? Alright? Gets nyo? Get Chicola Bells?
Yes.
Valiant?
Yes.
Get Chicola Bells mo? Alright. Naging ano na ako, from American drag queen to Filipino drag queen na. Get Chicola Bells, nag-gay lingo na si sir. Alright. Okay, so we're almost finished. Don't worry. I'm not going to, we're just going to talk about, I think, nutritional requirements for microorganisms. And hopefully you enjoyed the lecture. Okay. Next is among all bacterial endotoxin, which is not heat labile. All right. So among everything that we're going to talk about, the only exotoxin that is not heat labile, therefore it is heat stable, pinahirapan ko sarili, is staphyloenterotoxin. Pansinin niyo kapag ka nagkaroon ng food poisoning ang isang patient caused by caused by MRSA or whatever staphylococcus staph aureus or infection that person has, kahit gaano kadaming fever meron siya, hindi nawawala ang pagtatae, 'di ba? Yun yung dahilan kung bakit inaalam ng mga doktor sa atin kung anong nakuha ng pasyente. Among all exotoxins, which of the following is not heat labile? Siya lang kasi yung nakaka-survive sa init kapag ka nag-fever ang tao. Alright? Now, let's talk about immunologic characteristics. Now, when we say immunologic characteristics, one is protein, one is lipid. Which one do you think will be much more immunogenic? The lipid portion or the protein?
Yung lipid po.
The more immunogenic, sakaling kita. Immunology and serology. Sino ang mas una sino ang mas immunogenic? Kasabi ko lang kanina, kailangan ng protein carrier. Pag kailangan ng protein carrier ng isang substance, much better ang metabolism. And metabolism includes also immunogenic activities. 'Di ba? So kapag ka kumapit sa isang protein or pag ang isang protein ay isa ng toxin by itself, it's already toxic. It's already immunogenic. Kasi nga protein nga siya, di ba? Remember your discussions in immunology, right? Or in blood bank. What are the target organ or rather target antigens? Mostly they are proteins, di ba? Hypervariable region changes because of the amino acid sequences. 'Di ba? The hypervariable region of antibodies is the only reason why something is hypervariable is because the ends of these antibodies are linkages between the amino acid structures of their antigenic counterparts. 'Di ba? 'Di ba? Guys, kayo talaga ha. Nakalimutan nyo na yung immunology nyo. Kayo talaga magkakadikit lang ang immunology, blood bank at saka bacteriology. Therefore, this one is less immunogenic. 'Di ba? Yan o, less immunogenic or antigenic. 'Di ba? It can be neutralized by antitoxins. O 'di ba? It cannot be neutralized by antitoxins. Ano ang panggamot natin sa endotoxins? Sige nga. Anong panggamot natin sa endotoxins? Antibiotic. Kayo talaga, nakalimutan yun na kung paano ginagamot ang mga mikrobyo. Kayo talaga. Antibacterials. Kayo talaga. Oh, okay na kayo. Ready na kayo para mag-exam or hindi pa? Sino dito? Sinong honest dito na kaya na niya mag-take ng exam ngayon at papasa sa exam ko? Na gagawin kong, na pinrepare ko one question per day. Kasi pinag-isipan ko ng malalim. Pinagnilay-nilayan ko. Diba? So, hindi pa kayo ready. Aminado naman tayo. Alexis, ikaw, feeling ko, yung itsura ni Alexis, feeling ko confident. Hindi din. Hindi. Carry, pero carry ka naman. Nakikerry mo naman ang discussion. Nakikerry, okay. Tingnan natin kung gaano mo nakakarry. Kasi gagawa ako ng post-test next week, okay? So, makikita nyo kung gaano ako gumawa. Patikimin ko sila ng post-test, Sir Marco. Mga 10 items lang. No? Oh, see, he's probably playing. Okay? Online kasi siya eh. Charing. Doon sa nilalaro namin. Anyway, let's move on to the pharmacologic effects of bacterial exotoxins. One is, when we say pharmacologic effect, it is the process at which how the exotoxin is metabolized by the body. So a cytotoxin kills the host cell, an enterotoxin damages the cells of the GIT. What is the GIT? It's not igit in Bisaya, pero it does cause igit. But GIT stands for gastrointestine. Okay? Hindi yan igit ha. Sa Bisaya, I know it causes diarrhea, pero hindi yan igit. Okay? Huwag kayong ganon. Alright? Para madali lang maalala, ha? Kayo talaga. Sino yung mga ano dito? Sino yung mga Ilongga at saka yung mga Bisaya? They would relate. Igit. May makaka-relate. Uy, relate sila. Pero it does cause diarrhea. Nakakaigit. Okay? Okay? Next, neurotoxins, they impede the transmission of nerve or impulse. They impede nerve transmission or impulse. Okay? Yung mga axon terminals natin. Alright? So, ganon. Tawang-tawa siya doon sa ano, kasi igit ko si Nigel. Tawang-tawa doon sa igit joke ko. Where are you from, Nigel? Where are you from?
Bohol po.
Girl, kaunsa ka, day. Kapisti, day.
Ano
Naloka
sige?
ako, day.
Level ka, sir?
Ako naigit ako, day. Anyway, hindi ako Bisaya, hindi din ako Bisaya, hindi din ako Ilongga actually. My mom is from Bohol. I don't know, from Leyte, but I never really learned the language. I learned all the funny ones. Okay? Anyway, differentiate the toxins from the toxicity of toxins from endotoxins. So highly toxic. So which of the two do you think is highly toxic? Exotoxins or endotoxins? Which one? Sige, Nigel. Hula lang. Huwag ka mag-alala kahit mali or tama. Nigel.
Exo po.
Very good. Oo, kasi 'di ba? 'Di ba? Kakasabi ko nga, 'di ba? Protein sila mas potent kasi sila. Okay? Ito kasi kailangan pa ng carrier. Okay? Less toxic and less potent. But that doesn't mean hindi siya nakakaigit. Okay? Alright? Tuwan-tuwa si Nigel. It doesn't mean hindi siya nakakaigit, Nigel. Anyway, let's move on. What is the most potent bacterial toxin to man? This is a question from the PAMET quiz show. I don't know. Some people claim that they asked this in the BOC test banks. I just want to ask you guys what it is. It's actually Clostridium botulinum, the botulinum toxin from Clostridium botulinum. Okay? Botulinum. Remember botulism? Are you guys familiar with botulism? Right? Canned meat bacillus. Okay? It's the most potent. Okay? The most potent exotoxin. Okay? 1 gram of it can kill a person. Okay? 1 gram of the botulism toxin. But remember that the plastic surgeons and aestheticians use botulinum toxin in what particular beautification procedure? Sige nga, sinong gusto dito maging derma?
Botox.
Botox. Talaga, ay nako, talaga pag mga bayuta talaga nakakatuwa. I love it. Favorite na talaga kita, girl. Alam na alam talaga. Gusto mo yan? Gusto ko yan. Ganun. I love it. For the Barbie arms. I know. Girl, alam niya talaga. Talagang winner talaga sa akin to. Si ano. Favorite na talaga kita. Hindi ko na favorite ulit si Mark. Alright. Thank you so much. Mark, huwag ka magtatampo kasi ikaw hindi mo ako nai-impress katulad ng pag-impress ha. Binibigla ni ano yung pag-impress sa akin. Minsan binibitin ako sa impress, sa pagpa-impress. Okay, so nakakatuwa si Valiant. I love it. Minty fresh ha. Okay. Next, endotoxins from the body fluids and surgical instruments can be detected using an amoebic lysate derived from the blood of what animal? It's actually a question from the board exams or sorry, ACPI. Amoebic lysate, meaning you have to lyse the amoeba. And that amoebic lysate comes from horseshoe crabs. Okay? Blood from horseshoe crabs can be used. And the scientific name is Limulus polyphemus. Tama ba ako? Yeah, Limulus polyphemus. Diba? Limulus polyphemus. It's actually known as the Limulus test, if you guys are not familiar with that one. And the positive result is clumping. Okay? The positive is clumping. All right? So the amoebic lysate will react with whatever toxin whatever toxin is present in the cells or sorry, in the instruments. Okay? All right. Now, we're going to talk about nutritional requirements. Do we still have time? I don't think we have time. This is
So,
yeah,
two,
we
two
only
minutes na lang po, sir.
yeah. So,
Bale.
we're going to end our discussion here, but before I end the discussion, I'm going to ask a couple of questions to everybody. I prepared a set of questions for everybody. Now, I'm going to end sharing this one. Okay, so I'm also going to end our lecture in the yeah, I'm going to end. I'm also going to end the lecture in our live stream. It only lasted for 17 minutes, but I think that was enough for you guys to at least